All right, guys, today we're going to be talking about DNA replication. And for chapter 16, it's going to be really important that you're very comfortable with the information from Unit 1, Chapter 5, when we talked about the structure of both DNA and proteins. So a quick review. At the bottom, I have a nucleotide. A nucleotide is made of 3 parts, pentose sugar, a nitrogenous base, and a phosphate group. And the carbons can be numbered as we go around the sugar. So the 1' carbon is attached to the base. The 2' carbon tells us whether this is DNA or RNA, since there's a hydrogen there instead of a hydroxyl group. This is deoxyribose. The 3' carbon has a hydroxyl group. The 4' carbon is at the point, and then the 5' carbon is attached to the phosphate group. The next thing to keep in mind is that the 2 strands of DNA are anti-parallel to each other. So the 5' end, which would have a phosphate group on it, is exposed on one end, and the 3' end is exposed on the other end of the same strand. And then the other half of the DNA double helix is anti-parallel, meaning the nucleotides are lined up in the opposite orientation. They're flipped around. DNA replication is going to occur 5' to 3'. What that means is that growth of the new DNA is accomplished by adding new nucleotides to the 3' end. And the point where they hook up is the phosphate group on the 5' end of the new nucleotide hooks up to the hydroxyl group on the 3' end of the growing strand. The new bond that is formed is the phosphodiester bond that connects one nucleotide to the next in a polynucleotide. If we start in the middle of the chromosome, there's a point that gets pulled apart where the 2 strands of the parent DNA are separated from one another. This is accomplished by several proteins, including helicase, which separates the 2 strands, topoisomerase, which relieves supercoiling, single-stranded binding proteins that keep the 2 strands apart, and RNA primase, which we'll introduce in a moment. The point where the 2 strands get pulled apart is called the origin of replication. On either side of the origin is a replication fork. So for each origin, there are 2 replication forks. We're going to focus on the fork to the left. I've labeled the 5' and 3' ends of each of the strands of DNA, but I want to point out that every single nucleotide has both a 5' and a 3' end. So on the strand that's on the bottom, 3' is to the left and 5' is to the right, and that doesn't matter what side of the origin you're on. On the top strand, the 5' end is to the left and the 3' end is to the right, and that's true on both sides of the origin. I've zoomed in on the replication fork to the left, and the first thing that has to happen for actual DNA replication to start is the enzyme primase builds a short stretch of RNA called an RNA primer. This primer is synthesized 5' to 3', so new nucleotides are added to the 3' end. Next, DNA nucleotides are added onto the 3' end. Again, synthesis is 5' to 3'. The enzyme that does this is DNA polymerase III. Why is it DNA polymerase III? Because it was discovered third. On the top strand, synthesis proceeds into the replication fork, into that Y-shaped structure. DNA synthesis is always 5' to 3'. This top strand that is synthesized continuously towards the replication fork is called the leading strand. DNA synthesis always happens 5' to 3'. That's great for our top strand. But what do we do about the bottom strand? If we add new nucleotides onto the 3' end of the RNA primer, that goes back towards the origin, but doesn't allow us to copy the other DNA closer towards the replication fork. This other strand is called the lagging strand, and it's synthesized discontinuously in short fragments called Okazaki fragments. Separate RNA primers are put down. New DNA is synthesized by adding DNA nucleotides to the 3' end. Again, DNA synthesis always happens 5' to 3', and it's still accomplished by DNA polymerase III on the lagging strand. Next, the RNA primer is removed by the enzyme DNA polymerase I. It's both a helicase that removes the RNA primer and a polymerase that adds new nucleotides to the 3' end of the preceding Okazaki fragment. Meanwhile, RNA primase has put another primer on closer towards the replication fork. New nucleotides are added to the end of this RNA primer by DNA polymerase III, as previously. DNA polymerase I removes the RNA primer from the preceding Okazaki fragment and fills it in with DNA. To join these two pieces of DNA together, we need a different enzyme that's going to join the 3' end of the new Okazaki fragment to the 5' end of the old Okazaki fragment. The enzyme that accomplishes this is called DNA ligase. As the replication fork continues to open, there's room for additional RNA primers to be placed in. DNA is synthesized by DNA polymerase III. RNA primer is removed by DNA polymerase I, and new DNA is filled in by DNA polymerase I. And Okazaki fragments are joined together by DNA ligase. Here's your homework to test your understanding of this process. Draw what's happening on the other side of the origin of replication, on the other replication fork. I've drawn it here below with the 5' and 3' ends labeled. Which one is going to be the leading strand and which one is going to be the lagging strand, top or bottom, and which direction is synthesis going to go on each strand. Also, to test your understanding, draw the DNA with the opposite orientation. So draw it as I've shown here, with the 3' end being on the top strand on the left-hand side. One more thing to think about with regards to DNA replication. The black strands here are the old DNA, and blue is the new DNA that's being synthesized. So in this orientation, the top strand is a leading strand, and the bottom strand is a lagging strand. The leading strand can be synthesized continuously all the way to the end of the chromosome. But what happens on the lagging strand? DNA gets synthesized by DNA polymerase III. RNA primers are removed by DNA polymerase I, and DNA is filled in by DNA polymerase I. This happens to each Okazaki fragment, and the Okazaki fragments are joined together by DNA ligase, all the way up to near the end of the chromosome. But the RNA primer on the very end is unstable. That RNA will degrade, and there will be exposed DNA with no partner on the other side of the double helix. The consequence of this is each time DNA replication happens, the chromosomes get a little bit shorter on the lagging strand. One way organisms deal with this is by having extra junk DNA at the tips of their chromosomes. This DNA is called telomeres. It's structural DNA that serves a function of protecting the ends of the chromosomes, like the little plastic pieces on the end of your shoelaces that keep them from unraveling. But our chromosomes can't just keep getting shorter and shorter, or eventually they'll run into genes. This is one factor that contributes to aging. In other cells, specifically in the germline stem cells that give rise to the gametes that produce the next generation, there's a special enzyme called telomerase. Telomerase has a built-in stretch of RNA in it that gives room for one more RNA primer to sit down, providing space to make one more Okazaki fragment and copy the ends of the DNA. Telomerase is normally only expressed in germline stem cells to make sure that each generation has full-length chromosomes. However, in some cancer cell lines, they inappropriately express telomerase to allow those cancer cells to continue to divide immortally, practically, without any consequence of getting shorter and shorter chromosomes.